All right, homeotic genes, Looney Tune style. So we're going to talk a bit about the homeotic genes. We've been talking about these developmental maternal effect, gap, parallel genes, etc. In Drosophila, so we can kind of see how that applies to vertebrates a little bit. Oops. Let's kind of check out what we got going on here. So let me get my pointer out. Boom. So what's kind of cool about the homeotic genes is, so the most famous of them, I'll also refer to them as Hox genes. But there's actually, Hox genes are just one part of homeotic genes. There's some other para-Hox genes and things like that are kind of also involved in development. But for our case, we'll if I say homeotic, or if I say the Hox genes, we're just talking about homeotic genes in general. Anyways, these are developmental genes. These include the parallels genes and things like that. But what's really kind of cool about them is their location on the chromosome is conserved and also directly linked to their expression within the developing organism, which is kind of cool. So we have this antennapedia complex here that we have lab, DFD, SCR, which I think is scarecrow, ANTP. Oh, I have the names over here. ANTP is antennapedia, sex combs reduced is SCR. It's not scarecrow. There is a gene called scarecrow too. Yeah. DFD is deformed. Like I said, they just describe the phenotypes of what happens when you knock them out. And labial is lab. Anyways, so as you can see, their order on the chromosome is actually conserved to where they're expressed. And then we have this bithorax complex down here where we have UBX, which is ultrabithorax. ABDB is abdominal B, and ABDA is abdominal A. And you can see that even though this denotes that they're in different parts of the chromosome or on different chromosomes. I can't remember exactly. But the chromosomal regions containing these two complexes, so they are on the same chromosome, but they're just separated by quite a bit here. This is called the homeotic complex C, or HOMC. So you can see the expression of comparing. So this is also this is the developing embryo. This is where they'd be expressed on the adult fly. We can stain some of these. So we have engrailed stained in blue. Antennapedia is green here. Ultrabithorax is purple. So this is just kind of showing you can see their segments too, but just showing you how they can kind of map where these things are. But now let's talk about relaying these to not Drosophila, because we want to focus. I try to focus mostly on vertebrates, mammals, humans, because I know most of you watching are pre-med or pre-health, but sometimes the models helps drive home the point, right? So as you can see here, this is our HOMC group that we just talked about, where it's this conserved region. And if you look in mouse, we have the Hox genes, Hox A, B, C, and D. And these are all conserved. So there's gene duplication, right? So, or whole chromosome or genome duplication event. So they have multiple copies of some of these alleles. But anyways, you can see that these regions are still conserved, right? So lab is akin or a homolog to, or ortholog, I guess, to A1, B1, and D1 here. And you've seen there's some that have had mutations accrue in them, so those copies are no longer there, or they're no longer functional genes. They might have had their promoters messed up or a premature stop codon or something, right? Anyways, so you can see though that the order of these genes are still conserved. And what's also very cool about this too is if we look, the pattern of expression still is conserved, right? So this is whole evo-devo evolutionary development where way back when mouse and Drosophila shared a common ancestor, This was what the developmental patterning has been conserved throughout that time, right? So, but yeah, you see that all the way down to the tail here, we have these A13, C13, D13, which would be akin to the ABDB. Still, in this case, the posterior, the somates down here express it, and the tail section of the Drosophila express it. Very cool. Okay, so paralogs. I said orthologs. Paralogs are what they are. Let me make sure. Yeah, that's pretty good coverage of the topic. So here, this is kind of a schematic representation of the chick and mouse vertebrae pattern. So same thing, instead of Drosophila though, we're doing chick versus mouse. But what's interesting too is that the expression of these genes change the type. So what this is a picture of the vertebrae. Okay, I'm going to take a step back and explain this from the beginning. So this is a picture of all the vertebrae in a chick on the top and all the vertebrae in a mouse on the bottom. So some of this stuff is conserved, and the total number of vertebrae that are there is identical in I think all vertebrates. I think that stat's right. Even giraffes, which you'd be like, well, they've got to have 1 million more because of their long necks or whatever. But or maybe all amniotes. Maybe that's what it is. I'll have to double check that stat or that fact. Anyways, so it's conserved, but how they're what type of vertebrate they are is different, right? And that is based on these gradients and these Hox genes here, right? So you can see that chick and mice have the same number of occipital, but the chicks got way more cervical vertebrae. So the chick's on the left-hand side here, so you can see way more of that. They have way less thoracic compared to a mouse, which has a lot more thoracic. They have relatively the same lumbar and sacral, but the mouse has a lot more caudal than the chick does vertebrae. So these gradients, these homeotic gradients, they're not necessarily changing the number of the vertebrae, but they're changing what they should develop into, right? So they're not making more of vertebrae. They're saying, in this location, this one needs to be a cervical versus a thoracic or whatever. And that is all based on that positional information. So it doesn't make the positional information that, like hunchback and caudal and bicoid, it doesn't cause more things to grow. It just causes what is already there to develop into its specific identity, if that makes sense, right? So it's just giving information of what it should be, not that it needs to make more of something, right? Because it's already there. So it just gives positional or like identity information, right? So if we, like, for example, this is, let me get my notes here. So this is a wild type of a mouse, I believe. Does it say up here? It does not say. So this is a mouse. Look at all the vertebrae or the ribs. Okay. So we have our lumbar vertebrae here. We have a sacral vertebrae here. We have our T13 thoracic vertebrae here. And then if you were to knock out Hox 10, which is and the paralogs, so you have to knock out because there's multiple copies of it, right? So this is Hox 10 A C C D D, right? So there's 2 copies of Hox 10A, 2 copies of Hox 10C, 2 copies of Hox 10D. So they've knocked out all of them, right? Because it's diploid, so you have a mom and a dad copy, etc. Anyways, so you knock out all of them. you then turn what the lumbar vertebrae were into thoracic vertebrae. So they develop ribs, right? So before, remember, these are what the lumbar vertebrae looked like. But if you knock out all of that HOX10 gene, all the copies and the paralogs, then that positional information is still there. So is changed, and you no longer have lumbar here. You have thoracic vertebrae. So remember, by knocking out this gene, we're not causing a reduction, or if we expressed more of it, we wouldn't cause more vertebrae or less vertebrae. We're just changing the identity of the vertebrae, right? So no matter what we do with the homeotic genes, we're just changing the type of vertebrae, the positional information or the characterization of it, not the amount of them. So similarly, if we were to do, okay, this is, okay, so that, and then here, if we were to knock out HOX11, A, C, and D, that the sacral, which we had before, let's look at the normal, the sacral before, you can see here, those will then turn into lumbar vertebrae. So it's kind of hard to see the difference. You can see they have this kind of protrusion here on the lateral sides of the vertebrae. So if you knock them out, then that disappears and they look more like the lumbar. So again, we're not reducing the number. By knocking out these HOX genes, we're not reducing the number of vertebrae or getting rid of them. We're just changing their identity. So that's it for homeotic genes. I hope that's clear. It's really important that that's clear, that it's not a mutation that will knock stuff out. It just changes their information on what they should be. So again, thanks for watching. I hope you learned something, and I'll catch you in the next video.